to find out what happens next, right? Checkmate. Well, sometimes not so checkmate. Somebody gets a checkmate. Okay. So everybody understands that there's two divisions to immune system. There's two groups of players, specific, non-specific, innate, adaptive. We already started talking adaptive, right? And when we think adaptive, what should we think of? Specific. Give me some specifics on the specific. B cells, plasma cells, antibodies, T cells. Because we haven't really talked too much about T cells yet. Antigen presentation, antigen recognition, yes? All of those things, specific. So we left off talking about antibodies and how they worked. Do antibodies kill antigens? No. No. They do what? They tie them up, right? Neutralize them. Incapacitate them, sort of, so other players in this war can come along and get rid of them. Like phagocytes or cytotoxic T cells can come along and poke big holes in them. Complement can fix and poke big holes in things. And the antibodies are all going to help initiate those things. So the next part of the textbook, and of course you've read, so yeah, I'm just kind of reinforcing something you already know, because that's what you should have been doing this weekend, not watching SpongeBob. Cell-mediated immune responses. So we know that there's cells that are important for this whole immune response. And the first couple that they talk about are helper T cells and cytotoxic T cells. Now, the helper T cells have a certain cellular marker on them. And we talked a little bit about that, right? We talked a little about that when we were talking about some nasty little virus earlier on in the class. HIV virus. These T cells that are going to help with this immune response, I call them cruise directors of the immune response, are what we call CD4 positive T cells. That means that these cells, these T cells, have a specific cellular marker on them, and guess what the name of the cellular marker is? CD4. So these T cells are going to help get everybody organized in an immune response. This diagram shows us how everybody sort of, or at least the CD8 positive T cells, which are another group of T cells that are involved in immune response directions, know what to do. CD4 positive T cells have very specific markers, T cell receptors, that communicate with some of our specific antigens. We're going to start talking about those guys in a minute. Those are MHC antigens. What's another name for MHC? She's got a big mouth of granola, so she can't say it. Major histocompatibility antigens. And there's two groups of them. There's MHC1 and MHC2. And they're both going to work together, and we find them on antigen-presenting cells. These are the guys, remember I told you about how does everybody know what to do? Who does everybody know what to fight? And we were talking a little bit about antigen. How do I recognize antigen? No. How do I recognize antigen? Her face. What now equated to an antigen? What do we call that part of an antigen that we can recognize? It's an identifier. Antigenic determinant. Very good. So you can't eat anymore. So remember that little picture of the antigen, and there were 3 different A, B, and C antigenic determinants, and I talked about antigen and how do I recognize her in a crowd? Her face. That's her antigenic determinant. Well, that portion of an antigen is going to be presented with the help of MHC1 and MHC2 class proteins. Remember, we find them on dendritic cells, and we call the cells that are going to show everybody what they need to fight antigen-presenting cells. Now, the 2 different MHC classes are going to process antigens and their antigenic determinants a little bit differently and present them to different groups of cells. You following me so far? Guess who or which class of MHC is going to present antigens to the CD4 positive T cells in an immune response? That's right, MHC class 2. MHC class 1, and we're going to find MHC class 1 on everybody, pretty much everybody. MHC class 1 proteins, according to your textbook, are displayed by virtually all body cells except red blood cells. They're too busy carrying hemoglobin. They can't be bothered with anything else. So, MHC class 1, every single cell has the ability to present. And they are recognized by the cells of the immune system. When dendritic cells with MHC1 have encountered something foreign, like antigen, they're going to engulf that foreign something, chop off her head, or remove the antigenic determinant and put it out on the surface of the cell on that MHC class 1 protein. Class 1 is going to interact with the CD8 positive T cell. Now that T cell is then going to differentiate and go poke holes in things. It's a little specific, non-specific kind of action. He's going to poke holes in everything that looks like what? Antigen, or whatever the antigenic determinant was for whatever antigen we're talking about. Both of these groups of cells, CD4 and CD8, are going to clone very similar to what we talked about when we talked about B cells, when they encountered antigens. But the CD4 positive T cells are going to get everybody in order. Not only are they going to communicate with CD8 cells along with antigen presenting cells and MHC class 1, but they're going to coordinate their attack. They're going to replicate themselves and make more. And the CD4 positive T cells are also going to coordinate proliferation of B cells. Without the CD4 positive T cell, does anybody know what they're doing? No. It's kind of like, one of those old movies where everybody's running around banging into each other and falling on the ground, and nobody knows who's what and who's doing what or who to go after. You know what I'm talking about? I've got to change my material up because some of you youngsters have never seen anything like that. But that's kind of what an immune response is without CD4 positive T cells. So, differentiation, clonal differentiation of those T cells is what they talk about next. And who's going to get them started? The antigen-presenting cells, presenting 2 different classes of MHC. In this particular diagram, we see MHC being processed and put out onto the surface with antigenic determinants on it. And basically the difference between the 2 is the way they present and how effective they present. If you look at, and we're going to get there in a minute, but I just want you to look at page 789 if you've got it, we see how their communication happens between MHC class II and the CD4 positive T cell. So a protein, a antigen, something comes in, and that's what we're calling the endogenous antigen. It's broken down, as we know phagocytes will do, and then I'm going to take portions of it, and what do you think the portions are of an antigen that we are going to have to be able to recognize? What do we call those portions again? Antigenic determinants, yes. And they're going to be processed. They're going to be processed in the endoplasmic reticulum. And they're going to be docked with some of the MHCs. In this case, we're looking at MHC class I docking. The cell, that antigen-presenting cell, is then going to take antigen's head that I stuck on a post and do what? Bring it out and show everyone. Present it to the cells in an immune response. So we see here the plasma membrane of those cells, antigen-presenting cells, and the docked antigenic determinant on class I MHC. Excuse me. Class II MHC is going to take antigens, extracellular antigens, and pull them in, break them down, and process them. So it's a little bit more complex a process, but we're going to end up with the same sort of result. We're going to end up with what on the surface of these antigen-presenting cells? What's the little red doohickey? An antigenic determinant. In this case, it's going to be presented by class MHC2 proteins. So we're going to go through the same process. It's a little bit more complex. And MHC class IIs are very important in getting an immune response going. Everybody with me? What's the difference between 1 and 2? I'll give you a hint. Exogenous, endogenous. MHC class I is going to present what? Endogenous antigens. Something that's in the cell already. Exogenous antigens, what are those? We have to bring it in and process it, right? So MHC class I, MHC class II are going to help present antigens to the players in an immune response. Who are the players in an immune response? Our specifics and our non-specifics, right? Our specifics are going to be T cells, B cells. So when I dock with that dendritic cell that has a viral antigen, and again, think of antigen, what's being presented on MHC class II, antigenic determinant, is going to dock with immunocompetent CD4 positive T cells. What the heck does that mean? What's an immunocompetent cell? It recognizes self-antigens. This immunocompetent cell also recognizes when that self-antigen, MHC class II, is carrying something. And it's going to pay close attention. Once it figures out what it needs to fight, it's then going to clone, make more of itself. And some of the cells are going to go dock themselves and hang out in lymph nodes, waiting for the next attack to occur. And some of them are going to then go cause others to get a kick their butts in gear and get them moving in an immune response. You with me? So they talk in detail about T cell activation and antigen binding, and that's basically what's going on in this diagram. So the clones are being selective, figuring out exactly what they need to fight. recognizing what's being presented by the antigen-presenting cells, and then going off to stimulate others to do their job. So CD4 positive T cells, another name for them is what? Helper T cells. So they're going to bind with self, non-self complexes of B cells. And this is where I go tell the B cells what they need to do. They're going to release interleukins. Interleukins. What the heck are those? Didn't they stimulate B cell proliferation? Yeah, you're right. So these complexes are going to start releasing chemicals, interleukins and cytokines. That should ring a bell, right? What are they going to start doing? Causing more who to make some more white blood cells that I need, activate B cells, push them into cloning themselves, and causing them to eventually differentiate into plasma cells that are going to make antibodies specifically against these antigenic determinants that they've been presented. So, helper T cells in cell-mediated immunity are also going to communicate with the CD8 positive T cell. And CD8 positive T cells will become activated and then turn themselves into who? Yeah, cytotoxic T cells. Those cells are going to run around and poke holes in things with what? They're carrying a picture with them. Yeah, antigen. That antigenic determinant is now locked in. That information is understood, and now I know who I need to go fight. So helper T cell stimulates dendritic cell to express co-stimulatory molecules needed to activate CD8 cells. Dendritic cells can now activate CD8 cells with the help of interleukins secreted by helper T cells. So again, without the helper T cells, nobody would know what they needed to do. Coordinates the whole immune response. So they talk in your textbooks about the cytokines. Do you have to memorize them all? No. Interleukins, they are all part of the cytokine family. Do you have to memorize them all? No. But you do need to know that they are both responsible for causing stimulation of the cells in an immune response. Yes? The next thing the book talks about is the specific roles of both these players. Helper T cells. CD4 positive T cells. These are the cruise directors of an immune response. Okay, they are going to help, and this is kind of a little bit of repetitive information. They are going to help with organizing all of the different players in an immune response. CD8 positive cytotoxic T cells are going to go and help poke holes in things. Poke holes in things. How am I going to poke holes in things if I'm a cytotoxic CD8 positive T cell? Well, that's how. So I can create chemicals inside my cells. They're called perforins. And guess what they do? Correct. They poke holes in things. So in this diagram, we see the mechanism of action. Once they know what they're supposed to do and who tells them what they're supposed to do, CD4 positive helper T cells, once they become activated, they can then recognize their target. Their target is carrying the antigenic determinant that they were presented. So they can release these little perforin granules along with a sort of like glue for perforin. It's called granzymes. So these two chemicals can band together and form little complexes very similar to what we saw complement doing, and poke holes in the target cell or target antigen. Granzymes can then enter, and they act like proteases. Proteases. What's a protease? Protease. ACE, I'm thinking enzymes. Destroys proteins. So these little granzymes can then be released into the offending invader and start to degrade its proteins. And I got a big hole in the side of me. And do I stand a chance? No. So these are the cytotoxic T cells. So these cells all work together in an adaptive immunity to protect you from foreign substances. This is a lovely picture of a cytotoxic T cell attacking an abnormal or cancer cell. Cytotoxic T cells, CD8 positive T cells, are constantly looking for foreign presentation in the MHCs, or foreign MHCs in general. Why would a cancer cell be a target for a cytotoxic T cell? Yep, it's not made correctly. What do you mean it's not made correctly? Give me specifics, because all I can recognize is what? I recognize my markers. I recognize my MHC. I'm immunocompetent. I know what my MHCs look like. So on cancer cells, because their genetic material is altered, which results in proteins that are altered, cytotoxic T cells can recognize them as altered or foreign and attack them. This is going on constantly in your body. When cells reproduce themselves, especially cells that reproduce themselves at rapid rates, mistakes are made. And your cells, the cells of an immune response, are also constantly surveilling your cells for normal and abnormal. You constantly make abnormal cells. Not to scare you or anything, but you're probably making cancer cells as we speak. Why aren't you all going to go of cancer? Because your immune system is constantly surveilling the area. If a cell pops up, what am I going to do to it? I'm going to kill it. So why do we get cancer then? If we have such a wonderful surveillance system. Yeah, we might slip under the checkpoint, yes? And give me a chance to make more of myself. And cancer cells, one of the things we talk about when we talk about cancer cells is the fact that they don't follow any of these cell rules. They reproduce themselves at a very fast rate, and that when they start getting crowded, normal cells will stop. They don't care. They keep going. The problem with them is they take over all of the natural resources available for the healthy cells, right? And that's the problem with the cancer cells. But you constantly are surveilling and killing off these guys every day. Okay? And that's a cytotoxic T cell. Another wonderful diagram. So all of the players, and this diagram sort of ties everything together, all of the players in an immune response are on this diagram. Do you see them all? Okay, so who are the players? We have adaptive versus innate immunity. Specific versus non-specific. Okay? We have our antigen-presenting cells. We have our surface barriers, hopefully preventing anybody getting in. And then we have internal defenses like, what's that complex there? Complement. If somebody gets by, I'm going to poke a hole in you. Cytotoxic T cells, I'll poke a hole in you too. I just don't know what you are. I know that you don't belong. And then I get my specific immunity, right? And that, don't get confused, the purple kind of stops there. The beige comes over and follows down to be more specific in my immune response. So antigen-presenting cells engulf phagocytosis, process. Antigen-presenting cells will then, on the surface, show us antigenic determinants, MHC class 1, MHC class 2. MHC class 2 is going to communicate with who? CD4 cells. MHC class 1, CD8 cells. And everybody knows what's going to happen next. CD4 is going to get the ball rolling. CD4 positive helper T cells are going to help initiate or stimulate others in this immune response to proliferate and attack. So we see CD4 helper T cells, and we see this little arrow. Co-stimulation and release of cytokines that are going to stimulate who? B cells. Yes? B cells activated, make more of each other. Some of us are going to go become plasma cells and make what? Antibodies. We'll secrete them into the circulation and distribute them throughout the body. Antibodies are going to neutralize antigens. And who's going to come along and get rid of the trash? Some of our guys down there. Non-specific killers, macrophages, natural killer cells, phagocytes, neutrophils, mast cells. Yes? Once they see that complex, they know it doesn't belong. And what are they going to do? Eat them up, break them down, and spit them out. Where are we? What's naive, you think?
So they haven't seen anything.
They haven't seen anything yet. So once they get presented by antigen-presenting cells, it's going to activate them. They're no longer naive. They know what they're fighting. So they become what we call activated.
So once they see, then they can.
Correct. CD8 is going to also do the same, clone, create memory cells, activated cytotoxic T cells to go out and help with the killing process, poking holes in things. Phagocytes will come along and pick up the trash. See how everybody works together? Yes? Well, once we get past the presenting part, then we have specific orders. Does that make sense? So we go from naive to activated. And then everybody knows what they need to do. But again, CD4 is the one that's going to coordinate everybody, including the B cells, to actively pursue the antigen presented. We good? So why is it that I just can't, and they also talk a little bit about something called the regulatory T cell. It can actually inhibit things. It's one of those, not quite sure what you do. We've got to do a little bit more research, but we can't forget to write about you a little bit. Okay? So don't get too crazy about the regulatory T cell. We're still investigating that one. So why can't I just then, if one of my kidneys is sort of wearing out, go get extra parts at the extra parts store from just about anybody.
They have their own antigen.
Yeah, your histology won't let you get organ transplants. And that's what they talk about next in the book, organ transplants and prevention of rejection. Because of the way these guys are set up, because of how they survey, because of how they fight foreign substances in the body, you can't just go get any old kidney and slap it in there. What has to be similar in order for you to use someone else's kidney or something else's kidney? These proteins. The proteins on your surface of your cells. The closer I can get to replicating your proteins on the surface of the cells of the brand new kidney you want to slap in there, the better the chances are that your body won't attack it and see it as foreign. So there's 4 different terms they use with respect to transplantation. Autograft, that's the best one. How come? It's yours. It comes from you. I take it from one place and I put it on another. What's an isograft? Genetically identical individual. Does anybody in this room have a genetically identical individual? You do? You are a... Say who? You have an identical twin sister? Lucky you, she's got spare parts. Yes? You have to be an identical twin, though. You know the difference? Yeah, identical twins come from the same one oocyte fertilized by one sperm. And when the ball of cells starts to break off, something happens and we form 2 balls of cells. And then they just continue making 2 brand new people. We can also have 2 separate oocytes fertilized by 2 separate sperm. Are you genetically identical even though you're twins or you might be born at the same time? No. You're 2 separate developing individuals from 2 separate oocytes. So you're lucky you have spare parts. You can do an isograft. Allograft. What's that? And that's where most of ours are. Same species. So someone passes away or is killed in an automobile accident and donates their organs. What do we do with those good special organs? We can harvest them as long as we keep that person with blood flowing through their bodies for long enough not to have that tissue die, and somebody else might be able to use them. Somebody else with what? As very closely as possible matching antigens, including blood type, right? To those tissues.
What if it's, if the person's like an old cause or whatever, that matter during those type of stuff, so
Yep.
you have to be the exact same blood type?
Yep.
For this, not...
Yep. Xenograft. And we do this all the time too. We pick on baboons and pigs and all kinds of... See, we're very close to baboons and pigs. 792. So we can take grafts from other species. Anybody ever hear of anybody getting a mitral valve replaced? We take them from pigs all the time. I don't know about cow. Pig I've heard of. I don't know about cow. Baboon is another one. Okay, that's called the xenograft. But again, when you go to determine whether you can be a donor, what do we have to determine? Well, blood type 1, antigens, those are important because you have antibodies to those antigens floating around your circulatory system. Tissue type. We have to do a histocompatibility panel to see if your tissue antigens are similar to the patient in which organs you will receive. All right, so on page 795, there is a table, 21.5. And all of these guys have a nice little blurb on what their role is in an immune response. So cells and molecules of the adaptive immune response is a nice little take it down into small bits way of figuring out who does what and when in an immune response. Okay? Everybody with me so far? What's an immunodeficiency? Yeah. It's an immune system breakdown. And there's many different immunodeficiencies. One of them is an acquired immune deficiency syndrome, or AIDS, and it's acquired by patients who, patients who have what problem? Yeah. AIDS and HIV is different, right? Not every person with HIV or HIV positive is going to get AIDS. Well, eventually, but not right away. So acquired immune deficiency syndrome, or AIDS, is due to a decrease in number or lack of help from my who? My helper T cells. Why can't they do their job? Yeah, they're taken over. They're infected with HIV, human immunodeficiency virus. You know, sometimes it just hangs out and some of the cells can do their thing, but when they kick into action, they're definitely going to depress the whole immune response. So a lot of the drugs that are out there that try to prolong the lives of patients that have HIV are designed to try and stop those viruses from kicking in and killing off those cells, preventing them from doing their job. Viruses can hang out for a long time. A little scary factoid. As you get older, more and more of your genome is not yours anymore. It's what? Viruses. About 1 to 2% of your genome at your age right now is not yours. It's viral. Scary, isn't it? That's why they've been around a long time, and they're going to be around a lot longer after we're gone, because they know how to survive. They can hide out in your cells. Perfect place. I'm not going to kill my own cells, am I? If I hide out in here and wait, every time you replicate yourself, you replicate me. We're good. Anybody get cold sores? When do you get them? Yeah, but specifically. Something high in acid. How about stress? Yeah, it's changes in your body, stress. That's the virus saying, oh, this ship may be sinking. What do I need to do now? Make more of myself so I can spread to other ships. Yes? That's how viruses work. So, understand what an immunodeficiency is. Understand the difference between HIV and AIDS. Who are the cells involved with this nasty little virus? Who? T cells. Which T cells? Specific. CD4 positive T cells. Yes? Or helper T cells. Those are the cruise directors of the immune response, right? If I have don't have a cruise director, everybody's going to be running around on deck and not know what to do. What's an autoimmune disease? Yeah, you start attacking your own cells. Some of those guys just stop knowing what they're supposed to do for a living. It's kind of like they all of a sudden flunk kindergarten and they're out there. So autoimmune diseases, and there's many, many different autoimmune diseases, and constantly we're finding out that more and more diseases are autoimmune-like. 5% of adults in North America, 2/3 of them women, are afflicted with autoimmune diseases. So they give us a list. Multiple sclerosis, myasthenia gravis, Graves' disease, type 1 insulin-dependent diabetes, systemic lupus erythematosus, glomerulonephritis, and rheumatoid arthritis, to name just a few, are autoimmune diseases. Huh? Do I have to know how to spell them? No. It's It's a multiple choice test. You'll see it on there.
Why would arthritis?
Many arthritises are autoimmune. You start attacking your own joints. Rheumatoid arthritis is a nasty one, too. Okay, so many researchers around the world are doing research on autoimmune disease. What is it that causes these cells to break down or flunk kindergarten, let's say? We don't know for a lot of these diseases, and there's constantly research going on. Certain viruses can cause cells to sort of go awry. Certain cancers can be caused by viruses, causing cells to start doing abnormal things. There's a vaccine on the market, and women, you probably have heard of this one, Gardasil. And what am I vaccinating myself against? It's called human papillomavirus, because we know that this nasty little critter can cause problems with what? Cervical cancer. Okay, so again, researchers are constantly trying to figure out why cells do what they do, why cells change, why cells will suddenly attack self, and that's what autoimmune is all about. Some foreign antigens can actually be very close to resembling self-antigens, and that might be a way. But new self-antigens can also appear on cells because of changes in genetics. And who might change our genetics? We just talked about those buggers. Viruses, right? So that we start seeing new proteins on the surface. The other thing the book talks about is hypersensitivities. Allergic reactions. Cells creating antigens and antibodies, I should say, at high rates due to substances that are irritating or irritants. Allergies. Anybody have allergies? What do they call the little substances that get you going? Now, histamines are going to be released and try to fight them off. They're called allergens. So they're sort of like the antigenic determinant of allergies, similar in structure. And they cause what we call an allergic reaction. Now, allergic reactions typically happen very quickly. And one of the big problems with a very quick or acute allergic reaction is all of the things we talked about before with dilation of blood vessels and release of fluids into tissues can happen very quickly. Constriction of blood vessels. Not only blood vessels, but what else? Airways. Okay, so in this diagram, we see antigens or allergens that invade the body. Plasma cells are going to produce large amounts of antibodies. Those antibodies, IgE class antibodies. What's an IgE? Oh, she's flipping. Very good. Keep flipping. IgE. It's a monomer. Yep. Redness, swelling, all those guys come to the area. Histamines get released. Secondary responses. All the other guys are going to come in and try to fight off these nasty little allergens or antigens. Mast cells are going to release histamines. Histamines are going to draw more players to the area. But it's also going to cause problems. Too many histamines in the area can cause problems with other tubes in my body, like mucus, lined tubes. When you're going through an allergic reaction, allergy season, box of Kleenex is probably a very good thing to have with you, right? Because why? More mucus production because of these chemicals in the area causes blood vessels to dilate and become more what? Leaky. Causes edema in places, swelling, itchy, watery eyes. Everybody feeling it? Sometimes when you have an allergic reaction to, and I'm thinking of seasonal allergies right now, but you can have an allergic reaction to anything. Your face gets very puffy, eyes get watery, nose starts to run, especially if that antigen is making its way through your nasal passages. And then it can also cause what with the respiratory system, which is a big problem? Constriction of small respiratory passages that lead to the little air sacs where gas exchange takes place. That's not good. Decreased gas exchange because of increased resistance. We're talking airflow. We're going to talk those same kinds of things when we talk about airflow through the bronchioles and respiratory tree that we talked about when we talked about circulatory system. Okay? So they talk about some of the different immune responses. Anaphylactic shock, that's very bad. Shock, bad. Why? Yeah, things start shutting down to try and compensate for foreign substances and foreign invaders. Subacute hypersensitivities, and that's where the itchy skin comes in. Cytotoxic or type 2 reactions occur when antibodies bind to antigens on specific body cells. Immunocomplex or immune complex type 3 hypersensitivities. And that's when these allergens become more distributed throughout the body. And delayed hypersensitivity can happen as well. So sometimes you get exposed to something, and to actually show itself on the surface, it takes several days, maybe even 1 to 3 days, for the outward effects of a hypersensitivity to show itself. Contact dermatitis, we've all heard that. Has everybody heard that term before? Sometimes some people will go through these reactions when these allergens touch their skin. Some people are very sensitive to changes in their clothes soap, for example. Anybody in this class? Yeah, kids were like that. So we always had to buy the same type of clothes soap, and if we changed it, or they changed where their clothes were washed, they would get itchy rashes on their skin. So that's delayed hypersensitivities and allergic contact dermatitis. Developmental aspects of the immune system, don't worry about it. And yes, my friends, we are done. So, for your learning pleasure, please remove your clickers from your bag. Use, in this chapter, use your tables. They're going to narrow down the information for you so you don't get overwhelmed. One of my favorite diagrams, this one. If you can sit there and explain this diagram to yourself, you're in good shape. Okay? So where is this diagram? Yeah, on page 794 in your textbook. There it is. It's all the players doing what they're supposed to do. Yeah, a few things are missing. They don't really include too much on innate, but know those two. Okay? So, use your tables. Any questions? You ready? Wait a minute. Oh, don't look. Wait a minute. All right, which of the following trying, and you better get this right because you saw the answer at the bottom. Which of the following act as the first line of defense against foreign pathogens? No, I'm not going to go back. Learn to be observant. So my first line of defense, is 36 our number? Everybody clicked, who's got it? Who doesn't have their clicker today? Who's not going to admit they don't have their clicker today? Good, you all have your clickers. Have you all clicked? 38 is the number. What's the answer? The answer is D. So you four people didn't pay attention. Shoot. There it goes again.
Oh, yeah.
Yeah, it's at the bottom. All right, from current slide. Resume slideshow. Oh, sugar booger.
Where is it?
I know, it goes away. I don't know why it does that. Yes, you can. All right. The two intrinsic defense systems have many components. What's intrinsic? Which of the following cell types is not a contributor to specific immunity? It kills stuff, but it's not specific. Does that help? 5, 4. That's our number. What's the answer? The answer is A. Woo! We got some work to do, kids. What's a plasma cell? It was a B cell, and after it was specifically stimulated by antigen-presenting cells, it changed into a specific plasma cell that made specific antibodies. So 13 people picked that one. B cells, specific. They're going to get activated specifically. T cells, specific. You with me? Macrophages are derived from you want to make sure you got the right one? All right, we done? I want to make sure I get it right. Oh, shoot, wait a minute. Monocytes, B. Ooh. Okay, guys, we got some studying to do. Monocytes squeeze out of the circulatory system and become macrophages. Blank and blank are the most common phagocytic cells in the blood. The most highest in number phagocyte. We done? What's the answer? The answer is C. Good work. The next highest in number after a neutrophil is a who? Lymphocyte. Is it a phagocyte? No. Who's next? Yep. Okay? So see where that answer comes from? All right. What characteristic do all inflammatory chemicals share? Remember, all of them share this. Five, 4, 3, 2. What's the answer? All of them. They're not all histamines, are they? No. They don't all stop bleeding? No. They're not all secreted by macrophages? No. But they all cause vasodilation so that things can squeeze out. The answer is C. Histamines are inflammatory chemicals. That wasn't the question, right? All right. An increase in white blood cell number at the site of injury, which is characteristic of inflammation, is called... What's the answer? Increase in white blood cell number. Yes? B is the answer. The process by which phagocytosis, or phagocytes, excuse me, exit capillaries and enter injured tissue is called... the process in which they squeeze out of the circulatory system into tissues. What's the answer? D is the answer. See the difference in the question? Are we keeping you up? Sorry. All right, your hand has a scratch. What signals would indicate that the injury is inflamed?
Come on now. You should know this one right off the top of your head. I love it.
I'm missing 2 people. 5. There we go. You sleeping or something? Answer. The answer is C. Oh, yes! Love seeing that. All right, the basic mechanism employed by complement. Complement. What does it do? To fight bacterial infections is... Five, 4, 3, 2. What's the answer? C. What's complement? Proteins that band together and poke holes in things, right? There's a place on the antibody that's going to fire up complement. When an antibody is attached to an antigen, it can stimulate complement fixation. Does that make sense? So we can, ooh, yeah. Okay. All right, so complement is a group of proteins inside the plasma and has to be activated somehow. Normally they float around and do their thing and they're happy with the rest of the plasma proteins. But when things go awry, like foreign invaders and antigen and antibody reactions, there are places on the antibody that can cause complement to band together to poke holes in things. It's not just that, though. Other things can cause complement to band together. And just the presence of foreign substances, phagocytes gone crazy, can do that too. Yes. Membrane attack complex, MAC. Okay? And that complex is going to poke holes in cells. So in this question, it says, what is complement going to do to kill off bacteria? It's going to poke a hole in it, and that's going to cause it to burst or lyse. Yes? So sometimes you read a lot more into a question than the question is, right? Did I do the display thing? Yes. Yeah, okay. All right, what would be the body's response if the hypothalamus detected a pyrogen? Five, 4, 3. What's the answer? Answer is A. You don't want to, a pyrogen is going to increase body temperature, right? And that's what the response of the hypothalamus is. It re-establishes body temperature. Now, as a result, when you get too hot to release excess, you do sweat. But that's not what the question was, right? Which of the following materials or compounds would be the most highly immunogenic? It's a tough one. 5, 4, 3, 2. Oh. Well. It would be A. I prematurely did not stop that. Most immunogenic, anything non-self. A hapten needs what to become immunogenic? You have to add a protein. So if I don't have the protein to lock into a hapten, is it going to be immunogenic? No, so not necessarily that. Plastic, one of the reasons why we use plastic a lot. in medicine is because it doesn't have any what on it. There's no antigens on it. So we don't react. Some people do react to some of the chemicals in plastic, but for the most part, I can stick in an IV. I can stick in all of this wonderful plastic in you, and I'm not going to cause a lot of problems immunogenically. And self, anything. Are you going to react to self normally? No. So not self-antibody. Would I react to my own antibodies? I'd be in deep doo-doo if that happened, right? Okay, the presence of blank proteins make it possible for our immune system to differentiate between our cells and those that are foreign. So what proteins allow us to figure out those are mine and those aren't mine? Five, four. What's the answer? What's the answer? You think? We just talked about them, didn't we? Those are my recognition, my major histocompatibility. Yeah, those are mine. No, those aren't mine. Yes? Antigenic determinants are just the part of the antigen I can recognize. Right? Let's see what we got, I'm afraid. Whew. Yep, no. It's B. Okay, do you understand why? Do you understand now? Are we good now? Those are the proteins on the surface of my cells that are going to tell me that's mine and that's not mine. Yes? Okay, blank become antigenic if they attach themselves to larger proteins. Oops. We just talked about this 2 seconds ago. Five, 4, 3, 2. What's the answer? The answer is A. A. Haptens. Haptens. Remember, I need a protein in order to become antigenic, nasty, pain in the butt, cause an immune response. Yes? Lymphocytes are educated within primary lymphoid organs. The B cells are educated here, while the T cells are educated here. All right. What's the answer? The answer is A. Yes! 2! That's good. This is a tough chapter, and I got 2. That's good. During the clonal selection of B cells, those B cells with complementary membrane receptors to the invading antigen will differentiate into what cells? So if it's got the receptor that says, oh yeah, I recognize you, what does this cell do next? Five, 4, 3, 2. What's the answer? The answer is C. No, B cells don't become T cells. Never, never, never. I don't have the same cellular markers as a T cell, do I? I can't become a T cell, right? I should bring in a diagram. I think it's in one of my biology books, and it maps out all of the different proteins on the surface of a cell. It's totally mind-boggling how many there are. But those cellular markers define you as a cell. I am a cell because I have these markers on my surface. I can alter the shape of those markers and they might become deficient, but I can't like make new markers of other cells. Does that make sense? Whatever. Yeah. So just say yes. Humor me. All right. A patient has been immunized against the chicken pox. What type of immunity is this? Five, 4, 3, 2, 1. What answer? First of all, break it down. Is it natural or is it artificial? It's artificial. I got a shot at the doctor's office, so I can automatically eliminate A and C. Now I get a 50-50 chance of guessing it right. Is it passive, like nobody in an immune response did anything? No, it's active. It causes all of these cells to band together and do its happy stuff. So now what's the answer? D. The answer is D. You see? So you should do that when you take your exam. Passive would be if I gave you the antibodies to the antigen. Okay? So it's not natural because I went to the doctor's office to do it, and it's active because all of the players will create an immune response. And I'll create antibodies, and those antibodies will protect me from further invasion. That's why you have an immunization, yes? That's why you go and have a polio immunization, because we don't want you to get what? We don't want you to get polio. We don't want you to get sick. And if I have enough antibody, 10 times as much as what I need when polio gets into my body, is polio going to make me sick? No. Yes? No. No, it's not going to make me sick. Yes, do you understand my... whatever. In which class of molecules are antibodies classified? What kind of organic molecule is an antibody? There's another way to ask that question. I'm hoping for another 100%. Five, 4, 3, 2. What's the answer? The answer is D. I am a protein. That's how come I can have all those different shapes. Proteins. Antibodies are proteins. Which of the following best describes an antibody's mode of action, how it works? Five, 4, 3, 2. What's the answer? The answer is B. Very good. I tie them up and hold them immobilize, and tell everybody else to get rid of them. Mark them for destruction. Good, 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 good. Antibodies don't poke holes in anything. They can cause things to precipitate, clump up, but they don't poke holes. Who pokes holes? Complement, cytotoxic T cells, perforins, granzymes. No antibodies. Yes? When antibodies develop against a person's own cells, this is an example of mad thumbs. 5, 4, 3. What is that? It is autoimmune against self. Ooh, nice. 3 in 1 quiz. That is good stuff.
We should have to take a test.
So you're going to do really well on the test, correct?